en biotecnología de proteínas. Es un programa de acuerdo entre la Universidad Estatal de Washington y el NIH, que es el Instituto Nacional de Salud. En su tiempo libre, Eduardo disfruta de actividades al aire libre, especialmente ciclismo de montaña y otras actividades como trabajar en mecánica automotriz. Pues sin más preámbulo, les voy a dejar con Eduardo Sánchez. Gracias.
Muchas gracias, profesora, por esa introducción. The title of my research, like she mentioned, is Boosting Granulation on Upflow Anaerobic Sludge Blanket Reactor, and I'm working on Dr. Haluk Bayinol's research group. A little bit of introduction of where Washington State University is. It's in the state of Washington on the eastern side of the state, close to the border with Idaho. Upflow Anaerobic Sludge Blanket Reactor, or throughout this presentation, I'm going to refer to it as UASB, is used to high-strength wastewater, and by high-strength wastewater, I mean wastewater with contaminants at a higher concentration than domestic wastewater. That comes from livestock, crops, municipal waste, or food waste. And the basic design of this reactor is that the water enters the reactor from the bottom and is dispersed evenly throughout the granular sludge, or the microorganisms that remove that organic content from the water, and in the process of doing so, they produce biogas, which can be collected and used as an alternative source of fuel. What occurs inside a UASB reactor is the process called anaerobic digestion, which is a series of biological processes that use a diverse population of microorganisms to decompose organic material into biogas, and it's very important that it does so in the absence of oxygen. Like I mentioned, the wastewater comes from different sources like livestock, crops, municipal wastewater, or food waste, and enters this reactor or anaerobic digester, and the microorganisms remove the organic content from the water, cleaning the water, and on the same process producing biogas. Biogas is a mixture of gases. It's 55% to 7% methane and 25 to 45% carbon dioxide. Anaerobic digestion is a process that it doesn't occur in a single step. It starts with the wastewater or insoluble organic matter, long polymeric chains that through the process of hydrolysis are broken down into soluble molecules. For example, lipids turn into fatty acids, polysaccharides into monosaccharides, and proteins into amino acids. After that, acidogens, the acidogenic bacteria, turn those sugars and amino acids into carbon dioxide, hydrogen, and ammonia, which is a really important step because the other next two steps are going to be using these products as their main source of energy. Then we have acidogenesis, where acidogenic bacteria transform that organic acids into acetic acid, hydrogen, and also carbon dioxide. And the last steps, methanogenesis, where there's two types of methanogens. We have acetoclastic methanogenesis, that we use sugars and amino acids as the initial substrate, and it's around 60 to 70% of the methane production inside a UASB. And then we have hydrogenotropic methanogenesis that use hydrogen as a substrate. The characteristics of upflow anaerobic sludge blanket reactors that makes it be outstanding compared to other anaerobic digesters is that it's able to retain large amounts of biomass. And it does so by their design, because the wastewater goes from the bottom and the media is suspended, the granular sludge is suspended. So gravity is forcing it to stay down, preventing the biomass from leaving the reactor. Also, it has great interaction with the granular sludge. Therefore, we have short hydraulic retention times. which is under 9 hours versus 10 days their anaerobic digesters suffer from. Because the water is constantly mixing the granular sludge, it produces biogas, and those gas bubbles act as mixing. So we eliminate the need for introducing more or an external source of mixing, reducing operational costs. What I refer as biomass inside UASB is a small bean-shaped structure called granules, and they're formed by a process called granulation, which is a conglomeration of biomass resulting from the self-immobilization of anaerobic microorganisms. This has a lot of benefits because the structural strength allows the UASB to maintain higher amount of active material, and in that structure, we harbor the metabolic groups that are responsible for anaerobic digestion. Here is an image of a section granule under a light microscope that I was able to obtain in the lab. And if you see throughout the inside, it has different layers that we hypothesis that are the different distribution of microorganisms inside the granule. To further examine that, we took SEM images or scanning electron microscopy images of granules where we fixed them using the freeze-dry method to remove all the water inside the structure, and we section it along and obtain images along this yellow arrow. And we found that at 10 micrometers below the surface granule, there's not really a defined structure that we were able to observe. And compared to the literature, this relates to EPS, or extracellular polymeric substance, that holds the granule and gives it its structural strength. Imaging 250 micrometers below the surface, we start seeing some shapes, some structures more. And 350 micrometers below the surface, there's a different rod-shaped structure from the previous layer, and that we can relate it to acetogens being outside on the outermost layer, acetogens that produce acetate, hydrogen, and formate in the second layer, and methanogens from that produce methane, hydrogen, and formate in the middle or the inner layer. But because only by SEM images, we can't completely say that this is true, because we only see structure and EPS. So we need to do other tests to be able to understand how the microbes are arranged inside the granules. And to do so, we use microelectrodes. Microelectrodes is a really powerful tool, and it's used to study the structure of various biofilms with minimal invasion, because the electrode tip is around 10 to 30 microns. The invasion or the disruption of the structure is minimal. They're capable of measuring microscope gradients of oxygen, hydrogen, carbon dioxide age, redox potential, electron transfer rate, and therefore related to electron transfer mechanisms. This is a really basic microelectrode example, platinum microelectrode, where we have a glass rod that we pull it with heat, and then insert a platinum wire that is etched on KCN, and seal it with heat around the tip, so only a little bit of the etched platinum wire is exposed. And then we introduce an electrical connection to be able to control it with our equipment. Doing a literature search, or why we use microelectrodes, is because it allows improved understanding of the microbial arrangement and the sequential degradation of the organic matter inside the granules. And reading different papers is something that hasn't been done too much. Only a handful of papers have done microelectrode measurements inside anaerobic granules. First, we have this one by Sato in 2007, where they obtained oxidation reduction potential, methane, and hydrogen measurements in anaerobic granules. this was done on granules coming from a brewery. Then we have Yamaguchi in 2001, where pH microelectrode study showed an increase in pH, a higher depth within the granule, and that's because of the consumption of volatile fatty acids to methane. And then another paper by Sato 2012, where they obtained concentration profiles of methane, hydrogen, pH, and oxidant reduction potential to demonstrate the spatial distribution of the granules in their organized structure. The problem or the reason why we're so interested in understanding how anaerobic granules work is because UASB reactors have long startup times. Granulation under optimal conditions can take 2 to 7 months, which is a problem when you have large quantities of wastewater to be treated, and then you're still working on preparing your biomass. Also, temperature fluctuations. This is not too much of a problem on places where the temperature is mostly constant, but on places where the seasons are more drastic changes in the temperature, it affects the methanogenic performance up to 10 to 20 times on temperatures below 35 degrees Celsius. And improper granulations conditions led to impurity of biogas, where the sulfite presence caused by poor reactant conditions favor sulfur reducing bacteria, therefore compete with the methanogens, defeat the purpose of collecting that biogas and using it as an alternative source of fuel. To understand how the microorganisms work, we first need to understand the basics, which is the extracellular electron transfer. This is based on electron transfer from electron donors to electron acceptors, series of reactions. There's two main categories of extracellular electron transfer. The first one is mediated electron transfer, or MET, where electrons are transferred using redox mediators from formate and hydrogen. Or we have direct electron transfer mechanisms that during this presentation we're going to be referring to as diet, where electrons are transferred to electron-consuming microorganisms directly without any mediators. But there's also three types of diet. We have diet via conductile pili, where the electron-donating bacteria have elongated appendages that are related or called also nanowires that have metallic-like conductivity, and then connect to the other methanogenic archaea, and therefore electrons travel from one organism to the other. We have also membrane-bound electron transport proteins by C cytochrome, where the membrane are able to exchange electrons between microorganisms. And also we have via conducting material, where various conducting material like iron or carbon are added to the mixture to help or enable that transfer of electrons. There's a big discrepancy on which is the type of electron transfer mechanism occurring, but we do know that hydrogen production from fatty acids is only favorable at low hydrogen concentrations. And if we have low hydrogen concentrations, we have low methane production. So it points out mediated electron transfer as being an improbable electron transfer mechanism in anaerobic granules if we want to increase methane production. Digging through the literature, there's many community analyses done on anaerobic granules, and I chose this paper because this is the same. They community analysis on granules from the same place I'm obtaining my granule samples, which is a potato wastewater treatment plant. So it treats wastewater with high potato starch concentrations, and they did a community analysis on those granules and determined that 46% are acetylclastic methanogens and 46% are hydrogenotropic. So only half of those microorganisms are able to even consume the hydrogen and transform it into methane. which leads to the first hypothesis that diet is the main electron transfer mechanism inside anaerobic granules. And the importance of this is that if we understand how electrons are transferred through the different layers in the anaerobic granules, we will be able to manipulate the microbial communities and understand what each change we're doing is, how it's affecting the microbial community in UASB reactors. To do so, we need a couple of tools to enable us to obtain that information. And first, we need granules. So we take granules from Penford Food Ingredients in Richland, Washington, which is 4 hours from Pullman, Washington, where WSU is. Then, because we can't go there every other day to get fresh samples, we need to set up a bench scale UASB reactor where the granules are kept in their as close to their natural environment as possible, and we obtain granules from there for each experiment. So this custom-made UASB reactor, it's built out of PVC and polycarbonate tubes, and it consists of a mainlet and a plastic mesh to keep those granules from leaving the column if the pump is turned off or on. It also has 4 sampling ports at different heights of the column, so we are able to remove a limited amount of granules without disrupting the whole column and introducing air, because we have to remember that we want to keep it under anaerobic conditions, so in the absence of air. And it has a media outlet for the wastewater and a gas collection outlet at the top to collect the biogas. The operating parameters that we operate are the ones that the same place where we obtain the granules are operating at, which is a temperature in the mesophilic temperature range, which is 24 to 40 degrees Celsius, a pH around 11.3, hydraulic retention time of 4 hours, and inflow biochemical oxygen demand, which is how much material or organic content is in that water. Initially, it's 154, and at the outflow, our goal is to have around 4 milligrams per liter. We have 1,200 liters of autoclave potato starch media that we also obtain from the same plant, and around 100 milliliters of granular sludge in the column. These are conditions to simulate the same operation conditions that the plant is operating at, so we can ensure that the granules are as close to as possible to their natural environment. After the media exits the reactor, it goes to what I call a profile chamber, and this profile chamber is a chamber that also kept the granules under anaerobic conditions and in upflow, but we can position a single granule in it and study it, because we cannot introduce a microelectrode inside the whole column, because it's hard to choose a granule or a granule study for the microelectrode measurements. We put them in this chamber, and with the microelectrode here, we can then study the granule by keeping it in a fixed position without alternating or changing the granule structure, because we want their measurements to be as representative as possible of what the granules are doing in their natural environment. This is an actual picture of the profile chamber that I designed in the lab, which consists of a 50 ml Falcon tube with different connectors, and inside there's a microelectrode and the reference electrode, and on the bottom the counter electrode. First, we want to obtain hydrogen micro scale gradients inside the granules, and this will be done by measuring the hydrogen partial pressure based on the diffusion of hydrogen through a silicone membrane to apply an electrode and an internal reference electrode. So this is a little bit more complex microelectrode, and it's a compound electrode. So the two electrodes, the reference and the working electrode, are in a single glass casing, and it has a silicone membrane that is gas permeable. This, if we observe an increase in the hydrogen concentration, it will reveal the hydrogen production by acetogens. But if we see a hydrogen reduction or no hydrogen at all, it means that either the hydrogen is not being produced or how much hydrogen is being consumed by nitrogens, proving or disproving if mediated electron transfer mechanism is the main mechanism for transferring electrons inside granules. The next thing we hope to do in the lab, or we've done in the lab with this setup, is profiling the oxidation reduction potential gradients inside the granule. The idea for this is that if we measure the redox potential and monitor syntropic activity of the granules as they decompose the organic matter in the absence of oxygen. And we expect that oxidation reduction potential profile will show a slight decrease from the bulk media, so just the wastewater. As the microelectrode touches the surface of the granule, that oxidation reduction potential will decrease because there's less material to be oxidized inside the granule. So we want a gradient to show us that the granules are actually consuming something. This is a preliminary study that I did to test the anaerobic, the profile chamber with a microelectrode study, and we used a micromanipulator, which is like an extra set of hands that controlled has controlled increases of the distance, so we can always replicate it and know the exact distance the microelectrode is being moved or is inside the surface of the granule. And what we see here, it's just jumping back and forth, so it's noise. And a possible explanation for that is that in this case, we took longer to take the measurements because it's really hard to see a 10 micron glass tip inside dirty water. So if you take long to do the measurements, there's always slight vibrations of the granule. And as the microelectrode breaks through the surface of the granule, what is going to happen is that wastewater is going to diffuse through that same hole and cover the complete the microelectrode. So in this case, we took longer, and the microelectrode, the only thing that it measured was noise. So it was just bulk oxidation reduction potential, and that's why the variation was only 25 millivolt. So for further experiments, we need to do this measurement faster and reduce the flow of the water or turn it off just during the experiments. The next thing we're working on in the lab with the granules is obtaining conductivity and conductance. So conductivity, denoted by the symbol sigma, reveals the material capacity to enable the flow of free electrons. This is in units of Siemens over meter or 1/ohms times meter, but we cannot measure conductivity in an experiment. We measure conductance, which is uses the symbol G, and it experimentally measures in units of Siemens or ampere over volt or 1/resistance. What affects conductance is the material property, so conductivity, length, and area. So conductance takes into consideration the structure and the geometry of what we're studying, and then we cancel the area and we're able to obtain conductivity, which is a number and a parameter that we can compare it to different setups or different types of granule, no matter the size of the granule. The challenge with doing conductivity inside granules is that now you have 2 electrodes inside the granule under, or again, 10 to 30 microns in diameter electrode tip, and you also have to keep it in anaerobic conditions and keep that gap between the 2 electrode surfaces constant. So you need to use that electrode distance parameter as for the calculation. So like I mentioned, we're going to be using a micromanipulator and modifying that profile chamber that I showed you to be able to use 2 microelectrodes, and that's what we're currently working on. We're going to use initially potential range from 3 volts, from 300 millivolts to minus 300 millivolts. This is based on literature that they've taken conductivity measurements inside in granules, but what they do is they completely crush the granule on top of an electrode surface. And this gives us an idea of how to modify the parameters, but it doesn't show us how the microorganisms or how the conductivity changes throughout the structure of the granule. We're going to have a gate potential, which is the electrode source, the drain and the source potential divided by 2. And then the source drain potential, which is the drain minus the source. And we want to keep that around 50 millivolt. We'll be optimizing it as we continue doing the measurements and tests for increased sensitivity. The voltage of the source drain needs to be small enough that the conductive current changes linearly, because we're going to obtain the current and the voltage of the source and drain. The slope of that will be our conductance. So we need to plot those, and we want it to increase linearly. Not increase, only change linearly. It doesn't if it increases or decreases, we just want to test how it changes. And by quantifying direct electron transfer locally, we're expected to see that we're going to see an increase of diet-capable microorganisms as we go deeper into the surface of the granule, because if we expect more diet-capable microorganisms to be in the methanogenic area, we expect that conductance to increase as we go deeper into the surface. But not only understanding the direct electron transfer mechanism is important, now once we get that knowledge, we need to know what we're going to do with it. How we're going to affect or modify that electron transfer mechanisms to produce more methane. So there's 2 factors that control granulation and the quality of and performance. We have tailoring operation conditions, so modifying the temperature, the hydraulic position time, and the pH, but that's only limited. We also have the availability and growth of the microbial population in the slough. So if we control the granulation performance and we change that microbial composition, we possibly can modify or increase the conductance or conductivity of the granules. And we do this because studies have revealed that there's diet-capable microorganisms inside granules, like Geobacter or Methanosatia. And that's been done on granules from different industries, like brewery, alcohol production, and food and beverage. And they have found that there's a relationship between, they have found a lack of relationship between the electrical conductivity and the microbial community inside those granules. So this is a big question to be answered, because they have found microorganisms capable of doing diet if they're pure, but inside granules, there's not a relationship between how much diet-capable microorganisms are to the conductivity or the conductance. And this study from Kaisan in 2020, so it's pretty recent, they took 28 granular sludge from different industries, and 22 samples had a conductivity of 8 to 4.5 microsiemens per centimeter, and 5 of those samples had a conductivity of 9.9 to 39.2 microsiemens per centimeter, which is a big change or a big variation. And 3 of those samples are taken from the same brewery. So we can hear that there's an inconsistency on the amount of diet-capable microorganisms inside granules, and that's due to the granulation process. In this table, they listed the granule sample from left to right in increasing or from higher conductivity to lower conductivity. And we see that the one that has higher conductivity is the one with higher amount of Geobacter, but not necessarily that relates to the other sample. So the second in the line doesn't have a lot of Geobacter. And that's what we want to answer. So if we modify the reactor conditions, we can have more diet-capable microorganisms. And if that's the case, how will it impact the conductivity inside the granules? Because if not, it's just basically a waste of time. It's not increasing or changing the performance at all. which leads us to our hypothesis, a second hypothesis, where granulation process will be accelerated, and therefore we're going to have shorter startup time if we provide electrochemically active microorganisms where diet has been already promoted. And the concept behind this is that we're going to provide an electrode, and that electrode is going to be removing electrons from the surface, so removing electrons from the microorganisms. On those microorganisms that are capable of donating electrons will be enriched, will be promoted, and their growth will be promoted on the electrode surface. And we're going to do the same thing, but in the opposite way, on a separate chamber, where we are going to provide electrons to that electrode surface, and the microorganisms like methanogens that are able to convert carbon dioxide to methane are going to be attracted to that electrode surface because there's a high density of electrons and they're able to consume electrodes. So they're going to grow in this electrode surface and be enriched. And when we remove the biofilm from those two separate chambers, that capability and those microorganisms that are already able to exchange electrons are going to be at a higher amount or concentration than if we let this process occur naturally. So the methods for enriching biofilm, we're going to do anodic enrichment, so promote the growth of anode-reducing bacteria, and then in a separate chamber, do a cathodic enrichment, where we're going to promote the growth of methanogens that can use electrons to convert that carbon dioxide into methane. And then once we reach a steady state current or a maximum current, we're going to remove the microbes from that electrode surface and inoculate a fresh, clean reactor without any other microorganisms with the same setup. And the reason behind this is that by doing so, we're going to be ensuring that the anode-reducing bacteria is efficiently transferring electrons to the externally, and that the cathode microorganisms are going to be able to obtain electrons, convert carbon dioxide to methane. And once we take this in two different chambers, we're going to remove them once again, and then mix them in a reactor or in a new UASB column during during the granulation process. The system that I designed in the lab to enrich the biofilm consists of 3 electrode systems. And the reason why we do a 3 electrode system is that we can have a really accurate point of reference by using an electrode with a known potential. And therefore we can compare the value of the current to that. If we use only a 2 electrode system, we're only going to measure the difference in potential, not the actual number. The working electrode in our case, it's carbon felt, and the counter electrode is carbon fabric, and both electrodes are around 11 centimeters by 11 centimeters in size. And we also, because this is anaerobic reactor, anaerobic microorganisms, we purge the reactor by bubbling it with nitrogen to prevent any oxygen from leaking into the reactor. The reactor is around 6.5 liters, and 5% of that is homogenized anaerobic granules. And the reason why we do this is we're basically restarting granulation. So granules are we obtain granules already in their natural state, and we break them down into hopefully single cells to restart that process of granulation and be able to measure it. And then we use around 10% of potato starch wastewater in this system. This is an actual picture of a system that we use for that, which is a small bucket and a lid that is laser cutted with, it's a polycarbonate lid laser cut with the different fixtures are inlets for the different electrodes. And this is some preliminary data that we obtained from that system, where we use 10% autoclave potato starch wastewater 5% granular sludge, and we polarize it anodically, so a positive overpotential of 300 millivolts versus silver chloride, which is our reference electrode. And we added a carbon source of sodium acetate 30 micromolar at 2.5 days. So we added a carbon source here, and we saw that the microorganisms started consuming those or donating electrodes to the electrode surface around 5 days, which is the maximum current, and then it started decreasing as that electron source or carbon source was depleted. We do the same thing in the cathodic range, where we polarize it at a negative overpotential, so 100 millivolt, a little bit under. And this is different because we use, instead of anaerobic granules, we use mixed culture, because this was done before we were able to obtain the granule cells. It was just to test our setup and understanding of the data. And it took around 12 days to reach a steady state current. But this is not so representative of our setup or our goals, because this is bubbled with oxygen and we want to keep it under anaerobic conditions. So we're going to do the same experiment or replicate the same experiment, but bubbling with nitrogen and using anaerobic granules. What we expect out of this set of experiments is that if we promote the availability and growth of diet-capable microorganisms, it will allow them to exchange electrons faster. And if they do that, we reduce lag time. So the time it takes them to provide the maximum amount of exchange of electrons to the electrode, that will be reduced and will reach maximum current in a shorter period of time, reducing startup time. And the importance of this is that we understand the electron transfer mechanisms inside anaerobic granules, and it will provide insight on how those microorganisms interact with each other. It will also help us to explain how to promote the growth of the species to enhance electron transfer, because that's something that has never been done before, at least on mixed cultures. It has been done on pure cultures where they add some conductive material, but we want to do this in a way that the industry can also benefit from it and be able to controlly increase the concentration of the desired microorganisms, and also reduce startup time, because we've found the problem that where we obtain the granules from, sometimes they cannot give us too much samples, because if they have an issue in the plant and those granules die or are washed out, it takes so long to grow those granules again that they basically have to resort to buying the granules from other plants. So we want to speed up this granulation process to help maximize the potential of UASB systems. And I would like to thank my research associates and the graduate students in my lab that always help me with questions or giving me a second perspective when I'm running the experiments. Also, my funding, which comes for the project, is National Science Foundation, or NSF. And my personal funding, how I am able to pay rent and pay for my stuff, which comes from National Institute of Health, or NIH. And the two scholarships that I was lucky enough to be awarded with, which is the Research Assistance for Diverse Scholars, or the Achievement Rewards College for College Scientists, or ARCS. And this is a picture of our campus, which is really big and really nice at night. So I really encourage you, if you want, to go into the website and check out the pictures of the campus and also the different graduate programs. I checked, and there's around 73 PhD or grad student different programs in different departments. And in our department, it has around 63 PhD students working in a really wide range of fields, from catalysis to biofilms and wastewater treatment, like I mentioned. And now, if you have any questions, please feel free to ask questions. If you si quieren hacer preguntas en español, no hay problema también.
Está bien, Eduardo, muchas gracias, ¿verdad? Bien interesante el tema. Quizás, ¿verdad? Discutiste algunos aspectos que son bien específicos. La mayoría de los estudiantes son, ¿verdad?, subgraduados, así que si hay alguna duda, algún interés, alguna pregunta, por favor, puede formularla abriendo su micrófono o puede, ¿verdad?, escribirla por el chat. Yo tengo algunas preguntitas ya, ¿verdad?, que me gustaría. Cuando empezamos a hablar de gránulos, ¿verdad? Sí, el gránulo básicamente se fundamenta, ¿verdad? Tu research en gránulos, pero realmente mi pregunta es, ¿hay compañías que fabrican los gránulos o las plantas que producen biogás tienen una sección dentro del proceso específica para fabricar los gránulos?
Sí, hay compañías que si tienen exceso, tienen la capacidad de venderlo. Pero cuando uno empieza el proceso de UASB, la producción no están consumiendo la materia orgánica. Hay un proceso de aclimatación en ese sistema y entonces básicamente no está operando. Así que usa el mismo sistema para crear los gránulos. Se crecen o se forman dentro del mismo reactor.
Ok, o sea que los gránulos se forman dentro del mismo reactor, no es que tú o sea, es que pensaba como catalizadores, que tú compras el catalizador y lo metes, no. En el bioreactor tú primero aclimatas el bioreactor para que vaya dándose el proceso de granulación.
Correcto.
O sea que no es que se formen los gránulos porque se compacten como pellets en un proceso, no, ellos se van floculando, es como una floculación ahí dentro del tanque.
El proceso ocurre naturalmente, pero tiene que ser en unas condiciones diferentes a las de una planta operando en condiciones normales. Y muchas plantas no tienen la capacidad de tener dos reactores separados, así lo que tienen es un solo reactor. Y cuando empiezan el reactor, crecen o generan los gránulos. Y si hay algún problema, pues tienen que resultar a comprarlo de otras plantas que tienen un exceso, porque de tiempo en tiempo tienen que remover el exceso. Y ese exceso, pues lo venden a otras compañías.
Ok, muchas gracias. No sé
¿Sí?
si alguno de ustedes tiene alguna otra pregunta. Si no, yo continúo con algunas par de preguntas que tengo. Pueden escribir, ¿verdad? Si no quieren hablar por el
Sí,
micrófono.
tengo una pregunta.
Ajá.
¿Quién habla, por favor?
Es
Disculpa.
Shakira Martínez, ¿me escuchan?
Sí, le escucho.
Sí, sí.
¿Me escuchan? ¿Hello?
Sí, le escucho, le
Sí,
escucho.
Ay, sí, es que ahora a mala pata se me puso la señal mala. Si quieres puedes continuar en lo que yo veo que está pasando con mi internet.
ok.
Okay.
¿Alguna otra pregunta? Listo,
Ok,
yo voy a...
¿Hello?
Sí,
Ajá.
Ya se restableció, disculpa. Voy a hacer una pregunta acerca de como que tu proceso de, por ejemplo, como que ir a hacer PhD y eso, porque
Okay.
usualmente cuando uno va a solicitar y eso, hay algunas universidades que te piden específicamente como un essay de dónde tú te quieres enfocar. Cuando tú estabas en ese proceso y te piden ese essay, ¿tienes algo muy específico o tú también puedes hablar sobre algo que sea más broad, más general, o tiene que ser como que algo bien específico?
Pues depende mucho. Yo lo que hice en ese proceso fue, me metí a la página del departamento y vi lo que cada profesor estaba haciendo en las áreas que se enfocaba. Porque tú, cuando aplicas, tú aplicas a un programa, tú aplicas al programa, al departamento en general, pero como había cosas que yo decía, algunos campos no me interesaban, yo me enfoqué en creo que dos. Y fueron bastante general, porque en ese momento pues no sabía, solamente sabía lo que había leído en la página del departamento. Así, y di mi experiencia, di si uno tiene experiencia de un internado en que fue y dices por qué te gusta este campo y por qué te gustaría aplicar al campo de X o Y en este departamento. Y es importante que lo hagas con tiempo. Yo no estaba decidido si iba a hacer escuela graduada o no, y yo empecé a aplicar a mediados de diciembre. Y eso no es muy bueno porque muchas aplicaciones cierran en diciembre o la primera semana de enero. Así que yo no tengo mucho tiempo para leer eh, mi carta de lo que le llaman el Statement of Purpose, para leerlo y asegurarme que estaba de acuerdo a lo que ellos pedían. Así que el mayor consejo que te puedo dar es que si quieres aplicar a la escuela graduada, lo hagas lo más temprano, empiezas a escribir eso lo más temprano posible. Eso va a cambiar de acuerdo a la universidad que estés aplicando, porque no todas, hay universidades que no tienen algunos programas, así que tú quieres hablar y mencionar cosas que en ese departamento lo hacen, o cosas que te interesaría hacer si te aceptan en ese departamento.
Sí.
Pero no te preocupes mucho si no sabes muchos detalles o no tienes un conocimiento muy amplio de esos campos, porque ellos lo saben. Ellos saben que uno, pues, obviamente no está trabajando ahí. Y hay campos bien específicos. Yo nunca imaginé que el campo de Biofilm era tan grande cuando apliqué. Yo solamente hablé de que me gustaría usar estas aplicaciones para enfoques médicos y en la industria. Y ellos pues te contactan. Entonces los profesores te van contactando y ahí te entrevistan y ven lo que te gusta y te explican también el programa de ellos, lo que ellos están haciendo. Y ahí tú puedes como que tener una mejor idea de lo que de verdad están haciendo. Pero al momento de aplicar es bien difícil saber muy específico, a menos que también puedes ver los profesores y los buscas en Google y ver lo que ellos han publicado y más o menos tienes una idea del campo en que ellos están trabajando.
Okay. Eduardo, aprovecho la pregunta de Shakira. ¿Sabes si el Departamento de Ingeniería Química o en general la universidad eliminó el GRE? ¿No
Sí,
lo ha eliminado? Porque,
yo cuando apliqué no esta universidad no tiene el requerimiento de que tienes que coger el GRE. Y anoche volví a verificar los requisitos para aplicar al programa y sigue la misma política.
no lo tiene. Y el
No
TOEFL para los
tomarle-
puertorriqueños. El TOEFL para los puertorriqueños, ¿es obligatorio?
No,
El,
no.
tampoco.
Porque como ya uno viene con la ciudadanía americana, no te hacen tomar el TOEFL,
Okay.
en que
Okay,
ya
muy
uno
bien.
es residente. Así que esos
Muy
son críticas que me ayudaron a aplicar en esta universidad, porque cuando yo apliqué tarde, no había tomado el GRE, así que pues no podía presentar los resultados de mi GRE porque no lo tenía. Y esta universidad no tiene ese requerimiento. Hay algunos departamentos que sí lo piden, hay otros que no. Por lo menos el de ingeniería química y biomédica no pide el GRE. Y es importante que cuando uno esté aplicando, eh, uno pregunte, o cuando en ese proceso ellos te van a contactar, te van a enviar emails, uno pregunte las opciones de funding, de beca, porque el departamento mismo es el que te nomina. Yo no tenía idea de estas becas ni de funding, y es a través del mismo departamento que uno pues, le dice, mira, ya yo soy de Puerto Rico, y ellos empiezan a verificar, entonces ellos te nominan antes de llegar a la universidad para esa beca. Es importante uno preguntarle cuando empiecen a contactar a uno eh, la posibilidad de nominar a uno para eso, para diferentes becas, porque es algo que es bien difícil saberlo uno desde afuera de la universidad, y es algo bastante específico de cada universidad, de entre diferentes universidades. Hay diferentes procesos o becas que ellos tienen acceso que no necesariamente uno las encuentra así en Google. Eso es otra opción, uno las puede buscar. Pero la que a mí más me resultó y me dio mejores beneficios es a hablar a través del departamento. Y hay profesores que si ellos quieren que tú estés ahí, cuando ellos entrevistan y si ven que tú eres un buen candidato para trabajar con ellos, ellos mismos buscan la manera de atraerte a laboratorios, nominándote para esas becas y diferentes programas.
Okay. ¿Alguna pregunta, muchachos?
Pues mira, en ese caso, pues muchas gracias por aclararme eso. Yo por lo menos iba a empezar grad school el año que viene, pero voy a empezar un poquito como en spring, no en fall. Y en verdad que tenía esa duda porque en las universidades que estoy evaluando, pues usualmente piden, o sea, el letter of intent, o sea, dónde tú quieres
Correcto.
enfocarte y pensé que como que, Dios mío, o sea, yo tengo tanto interés y a veces como que, no sé, no, tampoco quiero overwhelm la persona que está encargado de ver la aplicación y no sabía si era necesario como que incluir tanto detalle y eso, sino tratar de hablar de por qué me interesa y eso, pero sin tener que escribir tanto.
Usualmente,
Realmente, ajá, disculpa, Eduardo, sí.
usualmente ellos te ponen un límite de cuán largo es esa carta. Creo que la mía era alrededor, el límite de mío era una página. Así que más o menos basado en eso, pues ya tú ves cuánto debes abundar en cada tema. Y si ves que son algunos campos que ellos no tienen acceso en ese departamento o no trabajan, pues no tienes que dar tantos detalles. Te enfocas en cosas que ellos, ese departamento, pues tiene acceso o que están trabajando en ese aspecto.
Y realmente ellos no buscan que tú seas experto en el tema, ellos quieren ver también otras cosas, o sea, cómo tú redactas, el impacto y lo interesante que es tu historia, porque básicamente tú lo que estás contando, yo me acuerdo que yo le decía a Eduardo, tienes que comenzar desde que niño tú jugabas, yo no sé si te acuerdas, jugabas, él es bien americano, porque pues él está aplicando a una universidad americana, así que yo lo que siempre recomiendo es búscate profesores que te corrijan el documento, o sea, que te aconsejen y que te puedan guiar, ya sean profesores que han estado contigo en internados afuera, ¿verdad? Pero busca como ese feedback, que profesores que ya hayan pasado por el proceso te den ¿verdad? Asesoría en realmente qué es lo que debe contener ese, porque ahí debe estar bien claro para qué tú quieres un PhD, para solo tenerlo, para ser profesor, porque amas la academia, te interesaría, o sea, es bien general, pero por lo menos tú muestras que tienes un plan de vida yendo a escuela graduada. ¿Está bien?
Ellos como el profesor Movil mencionó, ellos buscan una historia y a veces uno piensa, a mí me daba como vergüenza de algunas cosas y eso no, ellos no les va a interesar eso, pero sí, ellos buscan que tú seas, tienes un propósito, no un propósito, sino como que una idea de un plan de acción lo que vas a hacer, que no vas a llegar ahí a ver, déjame decidirme qué voy a hacer. Ellos buscan que tú tengas ese deseo o sea, de aventurarte y tengas un plan de acción. Obviamente ese plan de acción va a cambiar un poco cuando empieces escuela graduada porque uno pues otros conocimientos y descubre cosas que yo no tenía idea de esas cosas antes de aplicar. Pero ellos buscan esa capacidad tuya de decir, mira, tengo este interés, me gustaría hacer esto, y este es mi plan.
Sí, muy bien. ¿Alguna otra pregunta, muchachos? Eduardo, que tú sepas, programas de internado de verano que los muchachos puedan aplicar para ir a Washington, a Pullman, ¿verdad? Porque tú no estás en Seattle.
Sí, es Pullman.
Estás en Pullman, que es una ventaja, ¿verdad? Yo lo veo como una ventaja porque Seattle es de las ciudades más caras de Estados Unidos. Así que vivir en Seattle es un challenge. Así que tú estás en Pullman.
Y aquí, el pueblo de Pullman, lo que viven son 37,000 personas y yo creo que como 27,000 son estudiantes. Así que es bien pequeño, la criminalidad es bien baja, es bien seguro y como es una comunidad bien diversa de muchos estudiantes, pues uno se siente cómodo rápido cuando llega, no se siente como que uno es fuera de ahí o uno se siente intimidado por la gente, tú sabes, bien amable, bien seguro, un sitio súper seguro, la criminalidad, como dije, bajísima. Y programas de internado, no sabría decirte cómo han cambiado por lo de esta pandemia, pero si uno aplica con tiempo, es algo que yo no lo sabía, ellos te dan, creo que hasta $500 para venir a visitar.
Sí.
Eso es algo buenísimo porque uno puede, pues, ir a ver el sitio si le gusta y hablar con los profesores, porque como yo apliqué bastante tarde y ese año, pues, por el huracán María, la graduación se atrasó, pues las clases terminaron en junio y no tuve tiempo de ir a visitar. Pero eso es algo muy importante porque cuando uno visita y habla con los profesores, uno ya tiene una mejor idea de qué están haciendo, la personalidad de ese profesor, si se lleva, se va acorde con tu personalidad, porque eso también es algo que influye grandemente. Y todas esas cosas, si uno aplica con tiempo, puede beneficiarse de tener una mejor idea de lo que uno va a estar haciendo y dónde va a estar aplicando y qué profesores uno va a escoger.
Muy bien. ¿Alguna pregunta, muchachos? ¿No hay preguntas? Yo tengo una curiosidad. Tú mostraste, perdón, ¿alguien va a hablar? Tú mostraste unas imágenes de SEM, ¿verdad? De microscopía de barrido electrónico.
Correcto.
A diferentes profundidades. ¿Cómo o sea, tú partías el gránulo? ¿Cómo tú sabes a qué profundidad estás tomando la foto? Eso me causó bastante curiosidad.
Pues déjame buscar la foto. Pues o el proceso de fijar la muestra, yo traje un gránulo y en freeze dry, que es instantáneamente remueve el agua a la muestra. Y luego de eso, con una navaja bien finita, lo partí por la mitad para poder y ver por dentro. Sí, al uno usar una navaja, pues distorsiona un poco la estructura donde hizo el corte. Pero yo probé diferentes métodos si debería partir el gránulo antes de hacer la fijación o después, y los resultados eran los mismos. No vi una notable diferencia entre la calidad de la muestra. Y entonces, básicamente, esta es la parte expuesta donde hice el corte. Y estoy obteniendo imágenes aquí, en este punto, a lo largo de esa línea amarilla. Para ver las diferentes capas o estructuras dentro de la muestra.
¿Y la muestra la recubriste con metal, oro o algo así, o es suficientemente conductora para que salga bien la imagen? ¿O?
Sí, la cubrimos con oro.
La cubriste con oro. Listo.
Y no muchas universidades tienen un centro de microscopía y es una herramienta buenísima porque es una electiva, coges una clase y ellos te permiten acceso al centro, tienes 60 horas de acceso al centro y que lo puedes usar para tus propias muestras, no tiene que ser
Qué brutal. Te voy a enviar
a la clase de ellos, sino es solamente a tu proyecto y te da unas habilidades increíbles en cómo uno puede ver sus muestras y relacionar lo que uno está obteniendo en el laboratorio, en las muestras en los exámenes.
En esa misma dirección, ¿verdad? ¿Cómo tú sabías cuando tú penetras el gránulo? ¿Cómo tú sabes que estás, o sea, cómo tú controlas la profundidad para siempre medir a la misma profundidad o estar en la misma área? Porque si tú miras en esta imagen, claramente tú ves diferentes secciones, ¿verdad? Cada sección tiene diferentes microorganismos. ¿Cómo tú sabes que tú siempre estás penetrando en las mismas primero la misma distancia entre los electrodos, que es bien importante, y a la misma profundidad. ¿Cómo tú sabes eso cuando estás midiendo la conductividad?
Pues, según lo que yo he visto...
el setup tú tenías como un diagramita.
¿Este es el que se refiere?
Donde estaban las agujas ya para medir la conductividad en el interior del grano.
Ajá.
Ahí.
Pues ese es uno de los grandes problemas que he tenido que enfrentar, porque los gránulos no son todos del mismo tamaño, ellos cambian de acuerdo a su madurez de 2 centímetros a 3 centímetros, empiezan de pequeñísimos y más o menos en su estado maduro son de 2 a 3 centímetros. Así que si yo penetro un gránulo con un electrodo y lo puedo medir a 10 micrómetros, no necesariamente voy a estar midiendo la misma capa en cada gránulo. Así que va a haber un margen de error y uno lo que quiere ver es un patrón de, mira, más o menos en 100 micrómetros uno ve un cambio y esto entonces lo relaciona con las fotos que uno obtuvo de SEM y con las otras muestras.
Listo. Entonces vamos a dar por terminado, ¿verdad? El seminario. No sé si la profesora Elba todavía se encuentra y quiera decirnos algo. Yo le quiero agradecer personalmente a Eduardo, ¿verdad? Por regalarnos de su tiempo. Sé que está bien ocupado por los calificadores y todo. Gracias ¿verdad? Por hablarnos de tu proyecto, de las oportunidades que hay en tu universidad. Así que, nada, esperamos seguir en contacto contigo. Muchas gracias.
Muchas gracias.
Gracias a todos. Gracias a todos los que han asistido.
Yo estoy aquí, te había escrito en privado, Eduardo, pero de todas maneras aquí en público te agradezco mucho por, como dijo Omar, por tu disponibilidad de tiempo y por habernos presentado esta charla. Espero que podamos mantenernos en comunicación y te deseo mucho éxito.
Gracias. Y si necesitan los estudiantes que todavía estén en la presentación, si